Welcome back to the deep dive. Today we're leaving the relatively simple world of bacteria behind.
Yeah, saying goodbye to those little prokaryotic bags of enzymes.
Exactly. And we are stepping into a realm that is far stranger and, frankly, much closer to home than most of us are really comfortable admitting. We are talking about the kingdom fungi.
It really is a completely distinct universe. You know, when we study microbiology, we tend to just lump fungi in with bacteria because, well, there are both microscopic bugs that cause disease.
Right. They both grow on agar plates. They both make you sick.
Exactly. But biologically, they're worlds apart. Fungi are eukaryotes. They are the great decomposers, the recyclers of the planet, and occasionally they become these accidental tourists in the human body.
And when you say they are eukaryotes, let's just sit with that for a second. That means their cellular architecture is fundamentally the same as ours. They have a nucleus holding their DNA, they have mitochondria powering their cells.
Right.
They have a complex endoplasmic reticulum. They are essentially built on the same blueprint as human cells.
Which brings us to the central tension of medical mycology. It is what we call the target problem.
The target problem.
Yeah. If you develop an antibiotic for a bacterium, you have plenty of targets. I mean, bacteria have peptid glycan cell walls. Humans don't have peptidoglycan.
Right.
Bacteria have 70S ribosomes. Humans have 80S. So you can basically carpet bomb a bacterium with a drug without even scratching the human host. But with fungi,
It's like trying to bomb a house without breaking any windows, when the house is made of the exact same bricks as your house right next door.
that is a perfect analogy because we share so much evolutionary history with fungi.
Wait, we share history with them.
Oh, yeah. We actually split from the fungal lineage after we split from plants.
Wow.
So our metabolic pathways are uncomfortably similar. If you try to poison a fungus's DNA replication or its protein synthesis, there is a very, very high chance you are going to poison the patient's liver or kidneys in the process.
Because our machinery is nearly identical.
Exactly. This is why our arsenal of antifungal drugs is so much smaller than our antibacterial arsenal. And it's also why the side effects of antifungals are often so much harsher.
So if the inside of the house is the same, we have to look at the exterior, the armor. Because fungi do have a rigid boundary that we lack, right?
Correct. This is our saving grace. Human cells just have a squishy plasma membrane. Fungi have a rigid, complex cell wall sitting right outside their membrane.
And this wall is constructed from materials that do not exist anywhere in the human body.
Right. And this is exactly where we aim our weapons.
Let's construct this wall for the listeners.
Yeah.
Because if you are taking a board exam or just trying to understand how antifungal drugs actually work, you need to know these three specific components.
Absolutely. So visualize a multi-layered shield. The innermost layer, the part providing the real structural integrity.
Like the steel beams of a building.
Exactly. That is chitin.
Chitin.
Chitin is a polymer of N-acetylglucosamine. It is the exact same stuff you find in the exoskeletons of crabs and insects.
It's basically a crab shell wrapped around a cell.
Yeah, pretty much. It is incredibly tough, rigid, and absolutely essential for the fungus to hold its shape.
Okay, so we have a chitin skeleton. What is layered on top of that?
Over that, you have a thick matrix of glucans, specifically beta-1,3 and beta-1,6 glucans.
Glucans, so sugars.
Yes, these are long, complex chains of glucose molecules. They form a dense mesh that gives the wall its tensile strength. And medically speaking, this is a massive pharmacological target.
Oh, right, the echinocandins.
Exactly. If you've heard of the drug class echinocandins drugs like caspofungin or mycofungin, they work by inhibiting the enzyme that builds these beta-glucans.
The beta-133 glucan synthase.
You got it.
So basically those drugs stop the construction workers from laying the bricks, and the entire fungal wall just collapses.
Precisely. The fungus basically bursts under its own internal osmotic pressure.
Amazing. So we have chitin, we have the beta-glucan mesh. What's the third layer?
On the very outer surface, sticking out to interface with the surrounding world, you have mannins or manoproteins.
Manoproteins.
Yeah, these are proteins that are heavily modified with mannose sugars. They act as the surface ID card for the fungus.
And unfortunately for the fungus, that ID card is essentially a wanted poster.
It really is. Our innate immune system, our macrophages and neutrophils, have receptors that are specifically tuned to recognize mannins and glucans.
The pattern recognition receptor.
Right, PRRs. When a macrophage reaches out and touches a beta-glucan or a mannan, it knows instantly. It says, this is not self, this is fungus. Eat it.
So the cell wall is both their armor and their Achilles heel.
Exactly. It protects them from the harsh environment, but it heavily flags them to our immune system, and it gives us a vital target for our drugs.
Now, before we start categorizing the specific diseases, we really need to talk about the physical shape of these aliens.
Morphology is everything in mycology.
Right, because in the clinical lab, you aren't usually looking at DNA sequences or PCR right away. You're looking at shapes under a microscope.
Yes. We generally divide the fungal kingdom into two main morphological camps, the yeasts and the molds.
And this isn't just some trivial visual distinction. It tells you exactly how they live and how they reproduce.
Exactly. Let's start with the yeasts.
I always picture these as the solitary introverts of the fungal world.
That is a very fair characterization. Yeasts are unicellular. They are round or ellipsoid cells, usually about 3 to 15 micrometers in diameter.
So think of them like bacterial cells, but much, much bigger.
Much bigger. And the reproduction is quite elegant. The bud.
A balloon analogy.
Right. Imagine you have a large balloon, the parent cell, and you start inflating a smaller balloon right out of the side of it.
Just a little bubble popping out.
Yeah. That little bubble is called a blastoconidia. Eventually, it pinches off completely and becomes a brand new individual yeast cell.
Though sometimes they don't pinch off all the way, right?
Correct. Sometimes they stay attached and they form a chain of sausages. We call those pseudohypha. But the primary fundamental mode is budding.
Okay, so those are the yeasts, unicellular, budding, and then we have the molds. These are the social networkers.
Molds are multicellular organisms. They don't bud. They grow by extending these long tubular filaments called hyphae.
Hyphae?
Yes, and these hyphae constantly branch and interconnect with each other, forming this massive, dense mat called a mycelium.
So if you've ever seen that fuzzy green velvet growing on a forgotten loaf of bread in the back of your pantry, you are looking at a mycelium.
Exactly. You're looking at millions of intertwined hyphae.
And these hyphae are essentially just pipes.
They are tubes with rigid walls, and in many medically important molds, these tubes are actually divided into little compartments by cross walls.
Like rooms in a hallway.
Right. Those cross walls are called septa. But the really cool part is that the septa have pores in them.
So they aren't totally closed off.
No. The cytoplasm and even organelles can flow freely back and forth between the compartments. It's a very communal living situation.
Now, here is where the biology gets really, really interesting. Evolutionarily, some fungi realized that being stuck in just one form, yeast or mold, was limiting.
It's a vulnerability.
Right, so they developed the ability to literally shapeshift.
The dimorphic fungi. This is arguably one of the most sophisticated survival strategies in all of biology.
Dimorphic. Two shapes.
Yes. These are fungi that exist as molds out in the environment. So when they're living in the soil, decomposing organic matter, they are filamentous molds.
But when they enter a human host,
they completely transform into yeasts.
We have that classic medical school rhyme for this. Mold in the cold, yeast in the beast.
It's catchy and it's accurate. But let's look at the why. Why would an organism expend the massive amount of energy required to completely reorganize its entire cellular structure just because the temperature went from 25 degrees Celsius to 37 degrees Celsius?
It has to be defense, right?
Yeah.
It's a hostile environment in the human body.
It is entirely defense. Think about it. A mold is big, it's fuzzy, it's branching everywhere. It is very easy for the human immune system to trap it, surround it, and attack it. Its surface antigens are completely exposed.
It's a massive target.
Exactly. But a yeast is small, it's compact, it's streamlined, slippery.
Very. By shifting to a yeast form, the fungus can circulate easily in the bloodstream. It can hide inside our own cells. And critically, it presents a completely different set of surface antigens to the host's immune system.
So it's a camouflage tactic.
Yes. It drops its mold antigens and puts on a yeast disguise to confuse the host.
It puts on a disguise to rob the bank. That is terrifyingly brilliant.
And finally, before we dive into the actual clinical syndromes, we have to mention one more group. The dark nights of the fungal world.
The dark nights. You're talking about the dematiaceous fungi.
The dematiaceous fungi, yes.
That sounds like a villain group in a fantasy novel.
Well, they certainly look the part under the microscope. These fungi produce melanin.
Melanin, the exact same pigment found in human skin.
The very same. They synthesize melanin and deposit it heavily into their cell walls.
Which gives them a very distinct look.
It gives their colonies this dark olive, brown, or even jet black color.
And this isn't just for aesthetics, obviously. Melanin is highly functional. Absolutely.
Mhm.
In humans, melanin absorbs UV radiation to protect our DNA. In fungi, melanin is a major virulence factor.
How so?
It scavenges free radicals.
Huh?
When our white blood cells try to kill a fungus, they usually do it by blasting it with oxidative bursts.
They basically try to bleach it to death with hydrogen peroxide and superoxide.
Exactly. But if the fungus is covered in melanin, the melanin just absorbs all that oxidative damage. It neutralizes the bleach, it protects the fungus. It is literally wearing chemical armor against our immune system.
Incredible. So we have the players on the board. We have the unicellular yeasts. We have the filamentous molds. We have the shape-shifting dimorphs. And we have the armored dementiaceous fungi.
Those are our antagonists.
Now, we are going to use a very specific framework to navigate the actual diseases they cause. We are going straight down. We're going to follow the depth of invasion.
This really is the most logical way to organize medical mycology.
It matches the clinical reality perfectly.
Right. We will start on the very surface of the skin, then we will move down into the cutaneous keratinized layers, then we will burrow into the subcutaneous tissue, and finally, we will inhale our way deep into the systemic organs.
A journey to the center of the host. So, segment 1, the surface. The superficial mycoses.
These are the infections that honestly barely even count as infections.
They are really just colonizations.
Exactly. These fungi are loiterers. They just hang out. They live exclusively on the absolute outermost dead layer of the skin.
The stratum corneum.
Right. Or they live strictly along the outside of the hair shaft. They do not penetrate living tissue.
And because they don't break the barrier, the immune system largely ignores them.
Yes. You don't get fever, you don't get deep, angry inflammation, you get cosmetic issues, discoloration.
And the most common one, the one that probably a significant percentage of our listeners have actually had without even knowing the name of it, is pityriasis versicolor.
Pityriasis versicolor, caused by Malassezia species, typically Malassezia globosa or Malassezia furfur.
Now, Malassezia is a fascinating bug because it has a very specific dietary requirement. It is a lipophilic yeast.
It has a complete addiction to lipids, fats and oils.
It's the friend who only comes over when you order pizza.
In a way, yes. It naturally lives in the areas of the human body where our sebaceous glands are the most active.
So the chest, the back, the face.
Exactly. It feeds on the sebum we produce. It needs those long-chain fatty acids to survive. In fact, if you are a microbiologist and you want to grow Malassezia in the lab, you can't just use standard agar.
What do you have to do?
You literally have to drizzle olive oil over the agar plate, or the fungus will just starve and die.
That is wild. So it's hanging out on our chest, eating our skin oils. Why does it cause those spots? We call it versicolor because the skin literally changes color.
Right. So normally it's just a commensal organism. It lives in complete harmony with us.
It's part of the normal flora.
Yes. But under certain conditions, usually hot, humid weather, or if the person is sweating a lot, it overgrows.
And throws a party.
And as it metabolizes all those skin oils, it produces acidic breakdown products, specifically dicarboxylic acids, like azelaic acid.
Azelaic acid. That sounds super familiar. Isn't that an ingredient in over-the-counter skin creams?
It is. It's used in dermatology to treat hyperpigmentation.
Oh, because it lightens the skin.
Exactly. Azelaic acid inhibits tyrosinase, which is the key enzyme our melanocytes use to make melanin.
So the fungus is accidentally bleaching the skin underneath it.
Precisely. It's a chemical byproduct. So when you go out in the sun, your healthy, normal skin tans, but the little patches where the fungus is sitting and pumping out azelaic acid, they don't tan.
You end up with these pale, hypopigmented spots all over your back or chest.
Or sometimes they look pink or light brown, depending on your baseline skin tone, and a tiny bit of mild inflammation. Hence the name versicolor.
Now, for the visual learners, or anyone studying for the boards, describe the microscopic appearance of Malassezia. This is one of the most classic board exam images ever.
It is unforgettable. If you take a scalpel, scrape those little scales off the patient's back, do a KOH prep to dissolve the human skin cells, and look under the microscope, you see a very distinct mixture.
What's the mixture?
You see clusters of round yeast cells right next to short, stubby, curved hyphae.
And the mental anchor for this is?
The spaghetti and meatballs appearance.
Delicious. Spaghetti meatballs equals Malassezia. All right, moving from the chest to the hands, we have our next superficial infection, Tinea nigra.
This is a much rarer condition. It's caused by an organism called Hortea vernecki.
Now, I remember our dark nights from the intro. The demachiaceous fungi.
Yes. Hortea vernecki is one of them. It produces heavy amounts of melanin.
And it typically infects the palms of the hands or the soles of the feet.
Exactly.
And because the fungus itself produces melanin, the clinical lesion looks like a brown or jet black stain on the skin.
It does. And you can imagine this causes absolute panic for the patient.
Oh, I'm sure. A patient wakes up, looks at their palm, and sees a dark, irregular, rapidly growing, flat black spot.
Their mind immediately goes to the worst-case scenario. A colonisation melanoma, an incredibly aggressive skin cancer.
That is a terrifying differential diagnosis.
It is. But with Tinea nigra, the pigment is entirely inside the fungus, and the fungus is just sitting completely superficially on top of the stratum corneum. It's not in the melanocytes deep down in the epidermis.
So how do you prove it's just fungus?
You can often diagnose it by just taking a scalpel blade and simply scraping it off.
Just scraping away the stain.
Yes, or you treat it with a topical keratolytic agent like salicylic acid to peel the top layer of skin. The stain literally just washes away.
The relief that patient must feel.
It's immense.
Okay, finally, for the superficial layer, let's talk about piedra, which just translates to stone.
This is an infection strictly of the hair shaft itself. The fungus grows and forms a visible hard nodule that completely wraps around the hair.
And we divide this into two types based on color, white piedra and black piedra. White piedra is caused by trichosporon, right?
Yes, trichosporon species. These form soft, white, or sort of cream-colored nodules along the hair.
And they aren't super strongly attached.
No, they can be pulled off the hair shaft relatively easily with a comb. It's often found in tropical climates, and it typically affects facial hair, axillary hair, or pubic hair.
And black piedra.
That's caused by Piedrea hortae. This one is different. These nodules are hard. They feel like little chunks of actual coal cemented to the hair shaft.
So they are gritty.
Very gritty. If you run your fingers through the infected hair, it feels rough and bumpy. And unlike white piedra, the black piedra fungus actually penetrates the hair cuticle slightly.
So the nodule is firmly attached. You can't just comb it out.
Exactly. You usually have to shave the affected hair to get rid of it.
Okay, so that covers the surface loiters. Now let's grab our shovels and dig one layer deeper. We are entering segment 2.
Yep.
The cutaneous mycoses.
Now we are dealing with the dermatophytes.
And the key biological difference here is their metabolic capability.
Yes. The surface fungi we just talked about, they were perfectly content just living on the dead skin debris and the oils. They didn't really eat the destruction of the host.
But the dermatophytes do.
They do. They possess highly specific enzymes called keratinoses.
They're the keratin connoisseurs.
They literally digest keratin, and keratin is that incredibly tough, structural, fibrous protein that makes up our skin, our hair, and our nails.
It's what makes our skin a barrier.
Exactly. They are nitrogen scavengers. They break down the tough disulfide bonds in the keratin purely to get to the nitrogen inside.
And because they are actively digesting the physical structure of the host, the immune system finally notices.
It absolutely notices. This is where you start getting real clinical symptoms. You get inflammation, severe itching, scaling, erythema.
And this brings us to the classic clinical term, ringworm, which we need to clarify immediately for everyone listening, has absolutely zero worms involved.
It is a complete historical misnomer.
The medical term is tinea.
Right. Tinea corporis, tinea pedis, tinea capitis. The ring shape that people see on their skin is purely a result of the fungal growth pattern.
Walk us through that geometry. Why a ring?
A fungal spore lands on the skin. It germinates and starts growing outward radially in a perfect circle, constantly searching for fresh keratin to eat.
Okay.
As it expands, the tissue in the very center of the circle runs out of keratin food. So the fungus in the middle either dies off or goes totally dormant.
And without the active fungus chewing on the skin, the inflammation in the center dies down.
Exactly. The center starts to heal and looks relatively normal. But the leading edge, the outer border, is where all the active, hungry hyphae are aggressively chewing into new skin.
So the border is red, raised, scaly, and very itchy.
Right. That's the active border.
So clinical pearl here.
Mm-hmm.
If you are a physician and you are taking a scraping to send to the lab for a KOH prep, do not scrape the flat, clear center of the ring.
Never scrape the center. You won't find anything but dead skin.
Always scrape the angry red active border.
That is where the hyphae are. Always the active border.
Now, biologically, we need to break down the 3 distinct genera of dermatophytes. These are the 3 musketeers of skin fungus: trachyphyton, microsporum, and epidermophyton.
Knowing the difference between these three is incredibly high yield for medical exams.
So how do we actually tell them apart under the microscope?
We distinguish them primarily by looking at their macrocanidia.
They're large reproductive spores.
Yes, the shape and the wall thickness of these large spores tell you exactly which genus you are looking at.
Let's start with microsporum.
So, ironically, given the name microsporum, this genus is characterized by having huge, prominent macrocanidia.
What do they look like?
They have very thick, rough walls. They're often described as acuminate or spiky.
So they look like spiky footballs.
That's a great mental image, spiky footballs. And the key clinical association you need to know is that microsporum species, like Microsporum canis, often originate from animals.
Canis, meaning dog.
Right, or they come from the soil. And in terms of what they infect on the human body, microsporum infects the skin and the hair, but it rarely, if ever, infects the nails.
Okay, rough walls comes from dogs or cats, hits the skin and hair, skips the nails. Got it. Next up is trichophyton.
Trichophyton is the ultimate generalist. It is the most common cause of dermatophyte infections, and it infects everything: skin, hair, and nails. No tissue is safe.
And morphologically?
Its macrocanidia are very different from microsporum. They are long, thin, pencil-shaped or cigar-shaped, and they have completely smooth walls, no spikes.
Smooth, pencil-shaped microcanidia.
But here's the trick with trichophyton. When you look at it in culture, you actually don't see very many of those macrocanidia. What you see are millions of tiny little microcanidia clinging to the hyphae.
They look like little teardrops along the side of the brand.
Exactly.
Okay, and finally, the third musketeer, epidermophyton.
Specifically, epidermophyton floccosum. This is a major pathogen. Its macrocanidia are very unique. They are smooth-walled, but they are club-shaped.
They look like beaver tails.
Yes, beaver tails. And they are usually grouped together in little clusters of 2 or 3 at the ends of the hyphae.
And notably, epidermophyton does not produce any microcanidia at all.
None. And clinically, epidermophyton infects the skin. So it causes tinea cruris, which is jock itch, or tinea pedis, athlete's foot. And it infects the nails, but it does not infect the hair.
Okay, so let's lock this in. Trichophyton hits all three: skin, hair, nails. Microsporum hits skin and hair, but not nails. Epidermophyton hits skin and nails, but not hair.
That is the matrix you need to memorize.
Perfect. Now let's zoom in on one specific clinical presentation. Tinea capitis, scalp ringworm.
This is a really fascinating one because the clinical presentation and the diagnostic signs depend entirely on how the fungus physically attacks the hair shaft.
We have two main patterns here: the ectothrix infection versus the endothrix infection. Ecto meaning outside, endo meaning inside.
Right. Let's look at ectothrix first. This is classically caused by microsporum species.
What's the mechanism?
The fungal spores surround the hair shaft and form a dense sheath around the exterior of the hair.
So they are hugging the outside of the hair.
Exactly. Biologically, this severely damages the outer cuticle of the hair. But the really cool physical trick here, the diagnostic trick, is fluorescence.
The Woods lamp.
Yes. If a child has a bald, scaly patch on their head and you take them into a dark room and shine a Woods lamp, which is a UV light, on their scalp, the hairs infected with microsporum will glow a bright, uncanny fluorescent green.
It looks like something out of a sci-fi movie. Why does it glow?
Because the fungus produces a specific metabolite, a pteridine compound, and deposits it in that sheath around the hair. That compound is highly fluorescent under UV light.
So clinical anchor, glowing green hair equals ectothrix infection, which equals microsporum.
Exactly. Now contrast that with the endothrix pattern.
The inside job.
Yes. Here, the fungus, usually Trichophyton tonsurans in the U.S., invades the actual interior core of the hair shaft.
It bores inside.
And it packs the inside of the hair completely full of spores.
Like stuffing a tube full of sand.
Right. This completely destroys the structural integrity of the hair from the inside out. The hair becomes so incredibly weak that it simply snaps off.
And it snaps right at the level of the scalp.
Yes. It doesn't fall out from the root, it breaks flush with the skin.
Leaving a tiny little stub.
Which leads to the classic clinical sign, black dot alopecia. You look at the bald patch and it looks like the hair has been cleanly shaved, because all you see are these little dark dots, which are the broken stubs of the hair shafts filled with spores.
And importantly, if you shine a Woods lamp on a trigophyton endothrix infection.
Nothing. It does not fluoresce at all.
Okay, before we leave the cutaneous layer, I want to talk about a phenomenon that deeply confuses patients and, honestly, a lot of junior doctors. The UD reaction.
The dermatophyte reaction. This is the immune system chasing a ghost.
Paint the clinical picture for us. What does this look like in the clinic?
Okay, imagine you have a patient who comes in with a raging case of tinea pedis, severe athlete's foot. The skin between their toes is macerated, it's cracked, it's highly inflamed. The local immune system in the foot is actively fighting a massive fungal war.
Right.
Because of all that tissue damage, fungal antigens, the little pieces of the dermatophyte, are leaking into the patient's bloodstream.
So the alarm bells are ringing systemically.
Exactly. The entire immune system becomes hypersensitized to these fungal proteins. It's a systemic delayed type hypersensitivity reaction.
A type 4 reaction.
Yes. So suddenly, a few days later, the patient breaks out in these intensely itchy, fluid-filled vesicular lesions on the sides of their fingers or the palms of their hands.
And what goes through the patient's mind?
The patient looks at their hands and thinks, oh no, I was scratching my feet and I have spread the foot fungus to my hands.
Totally logical assumption.
Extremely logical. But here's the trick. If you take a scalpel, open up those blisters on the fingers and culture the fluid, or do a KOH prep.
What do you find?
Nothing. There is absolutely no fungus in the hand lesions. It is completely sterile.
It is purely a sympathetic inflammatory response.
Yes. It's an allergic reaction occurring in a totally different part of the body, triggered by the circulating antigens from the foot.
So how do you treat the hands?
You don't. Or rather, you just give them a mild topical steroid to calm the itch. The real treatment is curing the foot.
Because if you kill the fungus on the foot, the antigen source goes away.
And the hands miraculously clear up entirely on their own. It's a brilliant, if frustrating, example of how our immune response can sometimes be more confusing than the actual pathogen.
Incredible. Okay, let's keep going deeper. We've done the surface, we've done the skin and hair. Segment 3, subcutaneous mycoses.
Now we are crossing a major line. The previous fungi, the dermatophytes, they were completely content just chewing on dead keratin on the outside.
They stayed on the surface.
Right. But these subcutaneous fungi, they are invading living tissue. The deep dermis, the subcutaneous fat, the fascia, and even the muscle.
But they have a limitation. They are not aggressively invasive primary pathogens on their own, right? They can't just bore through intact, healthy skin.
No, they can't. They need an accomplice.
And the accomplice is trauma.
Exactly. The mechanism here is traumatic implantation. These fungi normally just live very peacefully in the soil, on rotting wood or on living vegetation. They have absolutely no interest in humans.
Until
a thorn, a wooden splinter, or a sharp stone physically punctures the human skin barrier and forcefully drives the fungal spores deep into the tissue.
the classic textbook case here, the one everyone learns in their first microbiology block, is sporotrichosis.
Rose gardener's disease.
The agent is Sporothrix schenki.
Yes. Sporothrix lives on rose bushes, on sphagnum moss, on hay, and decaying wood.
So the classic history is a gardener who is pruning their rose bushes, they aren't wearing thick gloves, and they get a deep prick on the finger from a rose thorn.
And that thorn inoculates the fungus right into the subcutaneous tissue.
Now, Sporothrix is a thermal dimorph, right? It shapeshifts.
It is. So out there on the rose bush, in the ambient environment, it's a mold. And if you grow it at 25 degrees Celsius in the lab, it forms these very delicate, beautiful, daisy-like clusters of Candida on the ends of thin hyphae.
Daisy-like clusters.
But once that mold gets injected into the warm, 37-degree tissue of the human finger, it realizes the environment has changed. It completely transforms into a yeast.
But the shape of this yeast is highly specific.
It is a cigar-shaped or fusiform budding yeast.
A little cigar. But what makes Sporothrix clinically unique, the thing that gives it away instantly in the clinic, is its travel itinerary. It doesn't just stay at the site of the thorn prick.
No, it creates a very specific chain reaction.
Walk us through the timeline.
So a few weeks after the initial prick, a small, relatively painless nodule forms on the finger. That nodule eventually breaks down and becomes an open ulcer.
Okay, primary lesion.
But then the fungus finds the local lymphatic vessels that drain that arm. It enters the lymphatics and it starts moving slowly upstream toward the lymph nodes.
Riding the curve.
And as it travels up the lymphatic channel, it gets trapped at various points, and you start seeing brand new nodules popping up in a linear line, tracking right up the forearm and the bicep.
It looks exactly like a line of red beads under the skin.
Yes. That pattern is called lymphocutaneous spread, and if you see a patient with an ulcer on the hand and a line of ascending nodules on the arm, your brain should immediately scream, Sporothrix.
And if you do a biopsy of one of those nodules, what are you looking for besides the cigar-shaped yeast?
You might see something called asteroid bodies.
Asteroid bodies. What are those?
It's basically a single yeast cell surrounded by a starburst pattern of pink eosinophilic material. That material is actually the host's own antibodies and immune proteins physically precipitating out and coating the fungus.
It's the Splendor-Hepley phenomenon.
Exactly. It's a visual marker of the immune system aggressively trying to wall off the yeast.
Fascinating. Let's move to the next subcutaneous disease. This one produces lesions that look like a horror movie prop. Chromoblastomycosis.
This is primarily a disease of the tropics and subtropics. It very often affects agricultural workers who are walking barefoot in the fields.
And again, it requires trauma. A cut from a tool, a scrape from a branch.
Yes. It's caused by a whole group of demaciaceous fungi. Remember, those are the dark, melanin-producing ones. Organisms like Malassezia pedrisoi or Phylophora varicosa.
What does this disease actually look like on the leg?
It is an incredibly slow, chronic, relentlessly progressive infection. Over a period of many years or even decades, the leg develops these massive, warty, hyperkeratotic lesions.
The textbooks always describe them as cauliflower-like.
That is the perfect description. They look like huge, dark, warty cauliflower florets growing out of the skin. They can become massive and highly deforming.
So how do you confirm the diagnosis? You take a biopsy of the cauliflower, and you look for a very specific hallmark structure.
The hallmark of chromoblastomycosis is the presence of sclerotic bodies deep in the tissue.
Sclerotic bodies, sometimes they are called miriform cells.
Right. These are actually clusters of vegetative fungal cells that get trapped in the dermis.
And because the fungus is demaciaceous, they have heavily melanized walls.
So under the microscope, they look like thick-walled, golden brown spheres.
The classic analogy is that they look like copper pennies.
Copper pennies. But if you look closely at those pennies, they are divided by thick internal septations. They have vertical and horizontal lines through them.
So they look like a pizza cut into slices, or a copper penny divided into quarters.
Exactly. If you see the words copper pennies or muriform bodies in a clinical vignette, you just mark chromoblastomycosis and move on.
Done. Finally, for the subcutaneous layer, we have to talk about mycetoma, also known as Madura foot.
This is perhaps the most devastating of all the localized subcutaneous infections.
What is it?
It is a chronic, slowly destructive, granulomatous infection of the deep tissues, almost always involving the foot. Again, starting from a barefoot injury.
What happens to the foot?
Over years, the foot becomes massively swollen, woody, and physically distorted. But the defining clinical feature is the development of deep, interconnecting tunnels draining sinus tracts.
Fistulae.
Right. These tunnels bore all the way from the deep muscle and bone right out to the surface of the skin, constantly discharging purulent fluid.
And if you look closely at that pus coming out of the foot, you find the diagnostic clue.
The granules.
Granules, like little grains of sand.
Exactly like grains of sand. These granules are actually dense, tightly packed microcolonies of the infectious organism.
Now, this is a massive point of differentiation. Mycetoma is a clinical syndrome, but it can be caused by two entirely different biological kingdoms.
This is critical. You have to distinguish between eumycetoma and actinomycetoma.
Eumycetoma meaning true fungal mycetoma.
Right, caused by true fungi like Mandarella mycetomatus. And then you have actinomycetoma, which looks clinically identical, but is caused by branching filamentous bacteria, organisms like Nocardia or Actinomedura.
Why is this distinction so incredibly critical? Why does the lab need to figure this out immediately?
Because the medical treatment is diametrically opposed.
Right.
If the lab looks at the crushed granules and says, hey, these are bacterial filaments, it's actinomycetoma.
Then you can treat it medically.
Yes. You treat the patient with long courses of antibiotics, drugs like Bactrim or streptomycin, and the prognosis is actually quite good. The lesions will slowly heal.
But if the lab says, these granules contain thick fungal hyphae, it's eumycetoma.
Then the situation is dire. Fungal eumycetoma is incredibly refractory to medical therapy. Standard antifungal drugs simply cannot penetrate the thick, fibrous capsule that the body builds around the fungal colonies.
The drugs just can't reach the fungus.
They can't. So medical therapy almost always fails.
So what is the solution for a fungal mycetoma?
Tragically, the only definitive treatment is often extensive surgical excision, debulking the tissue, or in advanced cases, full amputation of the foot. Yes. It is a terrible disease because it primarily affects impoverished agricultural workers in developing nations, and it leads to severe permanent disability.
That is a very heavy clinical reality.
Yeah.
But we must keep moving. We are entering the most dangerous territory now. Segment 4, The systemic invaders,
Yeah.
the endemic mycoses.
This is the major leagues. We are done with thorns and splinters and superficial skin scrapes. These pathogens do not need a traumatic introduction.
They enter through the most vulnerable, massive portal we have, the lungs.
We simply breathe them in.
These are the true, highly virulent thermal dimorphs. They are capable of causing serious disease in completely healthy, immunocompetent people.
Yes, they are primary pathogens.
And the overarching rule for this entire segment is geography. Geography is diagnosis.
Absolutely. These fungi are not found everywhere. They are highly restricted to very specific geographic regions and microclimates. If you know exactly where the patient has lived or traveled, you almost certainly know the burg.
Let's start our geographic tour in the American Southwest. The lower Sonoran Desert, Arizona, Southern California, New Mexico, parts of Texas, the land of coccidioids.
Coccidioides immitis and Coccidioides posadaceae. This is the agent of what is colloquially known as valley fever or San Joaquin Valley fever.
Talk about the environment.
It grows exclusively in the alkaline, semi-arid desert soil. The mold form in the dirt produces these very unique spores called Arthrochonidia.
Arthrochonidia.
What do they look like? They are barrel-shaped or rectangular. Think of them as a string of rectangular beads on a thread.
Okay.
As the mold matures, the little spacer cells sitting between the actual Arthrochonidia naturally undergo programmed cell death. They dry up and shatter.
Making the string incredibly fragile.
Highly fragile. So any slight disturbance to the desert soil, a strong gust of wind, a dust storm, an earthquake, or human activities like construction or dirt biking instantly shatters the hyphae.
And it sends millions of these little rectangular beads airborne.
And because they are so light and buoyant, they travel for miles on the wind. You inhale them, and they go deep, deep down into your terminal alveoli.
And then the transformation begins. And this is arguably the most dramatic shapeshift in the entire fungal world.
It really is.
Because it doesn't just become a simple little budding yeast like the others, does it?
No, it becomes a monster. It becomes the spherule.
The spherule.
A spherule is a massive, thick-walled sac. It is huge. It can be anywhere from 20 to 60 micrometers in diameter.
To give the listeners some scale, a standard human red blood cell is about 7 micrometers wide.
Right, so a spherule is easily 5 to 8 times larger than a red blood cell.
Which means?
Which means a human macrophage simply cannot swallow it.
Okay.
The primary defense mechanism of the lung, phagocytosis, is rendered completely useless simply by the sheer physical size of the spherule. It is immune to being eaten.
And what is happening inside this massive spherule while it's sitting safely in the lung?
It is undergoing rapid, continuous internal divisions by cleavage. The cytoplasm divides again and again, creating hundreds, sometimes thousands, of tiny little endospores inside the main sac.
The pressure is building.
Building and building. Eventually, the thick wall of the spherule ruptures, and it unloads this massive swarm of endospores into the surrounding lung tissue.
And each one of those tiny endospores can then grow into a brand new spherule.
Yeah.
It's basically a biological cluster bomb going off in the lung.
It is exactly like a cluster bomb. And the immune response to this cycle of growth and explosive rupture can be intense.
Now, what does this look like clinically?
Well, fortunately, about 60% of people who inhale the spores are completely asymptomatic. Their cellular immune system walls it off before it gets out of hand.
The other 40%.
They get a severe flu-like respiratory illness, high fever, cough, chest pain, extreme fatigue.
And there's a very classic dermatologic sign that comes with valley fever.
Yes, erythema nodosum.
The painful red lumps on the shins.
Right. The old term for this was desert rheumatism because you'd get the lung symptoms, severe joint pain, and these bright red tender nodules on the anterior lower legs.
Now, erythema nodosum isn't actually an infection of the skin, right?
Correct. It is a delayed hypersensitivity reaction. It's a sign that your cellular immune system is highly activated and is actively fighting the fungus.
So seeing those bumps is actually a good prognostic sign.
Paradoxically, yes. It means you are mounting a strong response.
But we have to talk about the severe end of the spectrum. Who is at high risk for disseminated disease? Where the fungus breaks out of the lungs and goes to the brain, the skin, or the bones.
Dissemination is the real danger. Obviously, anyone who is profoundly immunocompromised is at high risk. HIV patients, transplant recipients.
But there are also some very specific, somewhat surprising demographic risk groups for coccidioids.
Yes, for genetic reasons we still don't completely understand, individuals of Filipino descent and African American descent have a significantly higher rate of severe disseminated disease.
And there is one more, very specific risk group, pregnant women, particularly in their third trimester.
This is a fascinating bit of pathogenesis.
Why pregnancy? You'd think a young, healthy woman would be fine.
It turns out that coccidioides is actually biochemically responsive to human hormones. The fungus has surface receptors that bind human estrogen and progesterone.
Wait, the fungus has estrogen receptors?
Yes. And binding those hormones actively stimulates the fungus. It increases the rate of spheral maturation and endospore release.
So it's exploiting the host's own hormonal state to supercharge its replication.
Exactly. The high hormone levels in the third trimester basically throw gasoline on the fungal fire.
That is incredibly insidious. Okay, let's leave the arid desert and head east to the humid river valleys, the Ohio and Mississippi river valleys.
We are entering histoplasma territory.
Histoplasma capsulatum?
The geographic association here is very strong. Ohio, Mississippi, Missouri River basins. But there is also a very specific microenvironmental association you absolutely have to know.
It's all about the nitrogen.
Right. Histoplasma loves highly nitrogen-enriched soil, and what naturally enriches soil with massive amounts of nitrogen.
Guano. Bird and bat droppings.
Exactly.
So we are talking about specific high-risk activities, cleaning out an old chicken coop, demolishing an old abandoned barn where pigeons roost.
Or spelunking, exploring caves that are filled with bat colonies.
Now, a quick point of clarification. Bats actually get infected with histoplasma. They carry it in their gut and excrete the live fungus in their guano.
Right. But birds do not get infected.
Because their normal core body temperature is too high, right? Like 41 or 42 degrees Celsius. The fungus can't survive inside the bird.
Correct. The birds just provide the fertilizer. The droppings land on the soil becomes nitrogen-rich, and the histoplasma mold grows beautifully in that dirt.
Okay, so the mold is growing in the soil. It produces these very distinct stores called tuberculate macroconidia.
They look like medieval maces. Big, thick-walled spheres covered in spiky projections.
You sweep the barn, the dust kicks up, you inhale the spores, they get deep into the alveoli. What is the survival strategy for histoplasma? Because it's the complete opposite of coccidioids.
It really is. Coccidioids survives by becoming huge, a spheral. Histoplasma survives by becoming incredibly small. It wants to be eaten.
It is an obligate intracellular parasite.
Yes. The mental anchor here is histohides.
Histo hides inside the macrophages.
Right. The alveolar macrophage comes along, sees the fungus, and engulfs it into a phagosome. Normally, the macrophage would then fuse that phagosome with a lysosome, a deadly little bag of acid and digestive enzymes, to destroy the bug.
But histoplasma has a trick.
It does. It actively modulates the pH inside the phagolysosome. It secretes alkaline substances that raise the pH, neutralizing the acid.
And without the acid, the lysosomal enzymes can't function.
Exactly. It completely disarms the killing chamber. It turns a deadly acidic stomach into a cozy, protected studio apartment.
And then it just sits there, inside the macrophage, happily letting into tiny, narrow-based yeasts.
And it uses the macrophage as a free taxi service.
Right, because macrophages are mobile.
The macrophage eventually wanders out of the lung into the lymphatics and travels to the draining lymph nodes. Then it enters the bloodstream and travels to the spleen, the liver, and the bone marrow.
The reticuloendothelial system.
Yes, and the fungus goes right along for the ride, safely hidden inside.
This is exactly why in severe disseminated histoplasmosis, you see profound hepatosplenomegaly. The liver and spleen are massively enlarged because they are stuffed full of infected macrophages.
And you see pancytopenia, low red blood cells, low white cells, low platelets, because the fungus is physically crowding out the bone marrow space where those cells are born.
So visually, if a pathologist looks at a biopsy of the liver or the bone marrow under the microscope, what do they see?
They see a large macrophage that is absolutely stuffed to the brim with dozens of tiny little oval yeast cells.
It looks like a bag of marbles.
A bag of marbles. That is the classic histologic picture of histoplasma.
Okay, next stop on the geographic tour. The Great Lakes, the eastern seaboard, and overlapping a bit with the Ohio River Valley. Blastomycosis.
Blastomyces dermatitis.
The environment for this one is decaying organic matter, particularly rotting wood and leaf litter along the heavily wooded banks of waterways.
So think of a hunter or a forestry worker or someone walking their dog along a riverbank in Wisconsin.
They inhale the spores. The lung infection is similar in presentation, fever, cough, lobar infiltrates that look a lot like bacterial pneumonia. But the yeast form of blastomyces is totally distinct.
Yes, morphologically it is iconic.
There's no man.
The snowman. Blastomyces is a very large, thick-walled yeast cell, and when it reproduces by budding, the connection between the mother cell and the newly forming daughter cell is exceptionally wide.
It's a broad-based bud.
A broad-based bud. So instead of a little balloon pinching off a narrow stem, the two cells look like two huge snowballs stacked on top of each other, a figure 8.
Broad-based bud equals blastomyces. Now, clinically, blasto is notorious for a very specific type of dissemination.
It is the great masquerader, specifically of cancer.
It loves the skin and the bones.
It does. When it disseminates from the lungs, it often goes to the skin, producing these highly raised, heaped-up, warty verrucous lesions with irregular borders. They often have little microabscesses in them.
And what do these lesions look like to a dermatologist?
They look exactly like squamous cell carcinoma. They look incredibly malignant.
So the classic clinical vignette is a patient from the Great Lakes region who gets a biopsy of a suspected facial tumor.
Right. The surgeon cuts it out thinking it's advanced skin cancer. They send it to pathology.
And the pathologist calls them back the next day and says, good news, it's not cancer. Bad news, it's a massive fungal infection.
It's full of broad-based budding yeasts. Blastomycosis is a master of mimicry.
Finally, let's head south of the border. We are going down to Latin America, specifically the rural agricultural areas of Brazil, Colombia, and Venezuela.
We are talking about Paracoccidioides brasiliensis.
Paracoccidioidomycosis. That is a mouthful.
It is, which is why we often just say South American blastomycosis, though that's technically outdated.
Now, morphologically, this one might actually have the coolest, most recognizable visual in all of mycology.
It is spectacular. The pilot's wheel.
The pilot's wheel. Describe that.
So unlike blastomyces, which just puts out one single broad bud at a time, the parent yeast cell of Paracoccidioides sprouts multiple buds simultaneously all around its entire perimeter.
Like the spokes of a wheel.
Exactly. It looks exactly like the steering wheel of an old sailing ship.
Or, if there are only two buds at the top, people sometimes call it the Mickey Mouse head appearance.
Yes, Mickey Mouse ears. Multiple buds coming off a central parent cell. That is Paracoccidioides.
And clinically, there is a fascinating gender quirk with this disease that perfectly mirrors what we saw with coccidioids, but in absolute reverse.
Yes. Remember how coccidioids loved estrogen?
Right. It made the disease worse in pregnancy.
Well, Paracoccidioides absolutely hates estrogen.
Oh, really?
Yes. The natural human hormone beta-estradiol actually strongly inhibits the phase transition of Paracoccidioids.
What does that mean in practice?
It means that if a woman inhales the fungal spores in the environment, the estrogen in her bloodstream actively prevents those mold spores from transforming into the invasive yeast form.
It blocks the dimorphism.
Exactly. The mold gets stuck in its easily killable form, and the woman's immune system clears it effortlessly.
So women are basically naturally immune to developing clinical disease.
Essentially, yes, as long as they have normal estrogen levels. So the clinical epidemiology is overwhelmingly skewed. The ratio of clinical disease is something like 15-to-1, male to female.
It is almost entirely a disease of adult men, because men lack the high levels of estrogen needed to block the transformation.
Exactly. It is a stunning example of host-pathogen metabolic interaction.
When it does cause disease in men, what does it look like?
It primarily causes severe mucosal ulcers, the mouth, the gums, the nose, the larynx, very painful destructive lesions in the upper airway, along with cervical lymphadenopathy.
Okay, we have covered the geography, we've done the true pathogens. Now, for the final major segment, we need to turn our attention inward. We need to talk about the traitors.
Segment 5, the opportunists.
This is a totally different game. The endemic fungi we just discussed, they are rugged outlaws. They live in the dirt, and they can attack a perfectly healthy sheriff.
Right. They are primary pathogens.
But the opportunists, they're cowards. They are low-virulence organisms that absolutely require a broken, compromised host before they make a move.
They need the shields to be completely down.
And the undisputed queen of the opportunists is Candida.
Candida albicans. It is by far the most common fungal pathogen in humans. But here is the major conceptual twist for students. You don't usually catch Candida from the environment.
No.
It is already inside you. It is endogenous. It is a normal, healthy part of the commensal microbiome of the human mouth, the entire gastrointestinal tract, and the vagina.
So if it's supposed to be there, what turns a peaceful roommate into a deadly systemic killer?
A sudden, drastic shift in the ecological or immunological balance. And there are three main triggers we see in the hospital.
What would be number one?
Broad-spectrum antibacterial antibiotics.
The collateral damage.
Exactly. Candida is constantly competing with millions of normal bacteria for food and real estate in your gut and mucosa. If a doctor puts a patient on massive doses of antibiotics, it wipes out all the normal bacteria.
And suddenly Candida has an empty mansion and a Philly stock fridge all to itself.
So it massively overgrows. This is what causes oral thrush, the white plaques in the mouth, or vaginal yeast infections after taking antibiotics for a UTI.
Okay, trigger number two.
Barrier breakdown, intravenous lines, central venous catheters, Foley catheters, major abdominal surgery, terrible burns.
You are physically providing a highway past the skin barrier straight into the sterile bloodstream.
Right. Candida lives on the skin and the mucosa. If you push a plastic tube through the skin and leave it there, Candida will happily crawl right down that plastic and enter the blood.
Candidemia.
Yes. Catheter-related bloodstream infections are a massive cause of morbidity.
And trigger number three.
Profound immune suppression. Specifically, neutropenia.
Neutrophils are the main defense against Candida.
They are the frontline soldiers. If a patient is undergoing harsh chemotherapy for leukemia and their white blood cell count drops to 0, Candida will simply cross the gut wall, enter the blood, and seed every organ in the body. The liver, the spleen, the eyes, the heart valves.
Disseminated candidiasis, it's incredibly lethal. Now, biologically, Candida is also a shapeshifter, but it is not a thermal dimorph, right?
Correct. We call it polymorphic. It can change its shape regardless of the temperature, depending entirely on environmental stress.
And it has three forms.
Yes. It can be a simple budding yeast. It can form pseudohyphae, those elongated chains of sausages. Or, under the right conditions, it can form true hyphae.
True. Branching multicellular tubes.
And this is the crucial point. The transition from yeast to true hyphae is actually its active invasion mode.
Explain that.
The yeast form is great for just floating around in the bloodstream or colonizing a surface. But when Candida wants to actually invade an organ, to bore through a tissue plane, it elongates into a rigid hyphal tube.
It essentially turns itself into a microscopic spear.
Exactly. It physically punches through the human cells.
And we actually exploit this ability in the microbiology lab for rapid identification, the germ tube test.
This is a classic lab technique. If you have a yeast growing on a plate and you want to know if it is Candida albicans or some other harmless yeast, you take a tiny sample and you put it in a test tube containing animal serum.
Usually fetal bovine serum.
Right. You incubate it at 37 degrees Celsius for exactly 3 hours. The serum strongly stimulates Candida albicans to start invading.
It thinks it's inside a host.
Yes. So the little round yeast cell begins to sprout the very beginning of a true hypha. It forms a short, thick, straight tube coming right off the main circle.
It looks exactly like an old-fashioned hand mirror, a circle with a straight handle.
That is the germ tube. And if you see that under the microscope at 3 hours, the test is positive. It is definitively Candida albicans.
Now, before we close out Candida, we absolutely have to mention the new nightmare on the block, the organism that is keeping infectious disease doctors up at night.
Candida auris.
This bug wasn't even known 20 years ago.
It was only first identified in 2009, isolated from the ear canal of a patient in Japan. Hence the name auris for ear. But since then, it has aggressively swept across the entire globe.
It is causing massive, unstoppable outbreaks in intensive care units everywhere. Why is it so terrifying?
For two major reasons. First, its resistance profile. Candida auris is frequently multi-drug resistant.
Meaning standard antifungals don't work.
Right. Some strains are resistant to azoles, like fluconazole. Some are resistant to the echinocandins. And terrifyingly, some strains are pan-resistant. They are resistant to azoles, echinocandins, and amphotericin B.
We literally have no drugs left on the shelf that can kill those strains.
None. It's a return to the pre-antibiotic era for those unfortunate patients.
And the second reason it's a nightmare.
It is its environmental persistence.
Most Candida species die pretty quickly if they dry out, right?
Yes, Candida albicans does not survive well on dry hospital surfaces. It needs moisture.
But Candida auris.
Candida auris is different. It can survive and persist on totally dry abiotic surfaces for weeks or even months.
I mean rails.
Bed rails, privacy curtains, vital sign monitors, floors, the walls of the hospital room.
So it acts more like MRSA or C. diff than a standard yeast.
Exactly. It colonizes the skin of patients, they shed it into the environment, and it is incredibly difficult to completely eradicate from a facility once it takes hold. Standard hospital disinfectants often fail. You need specialized sporicidal cleaning protocols.
So Candida is the enemy within, and increasingly the enemy lurking on the bed rail. Now, we could spend another hour talking about Aspergillus, the ubiquitous environmental mold that violently invades the blood vessels of neutropenic patients.
Causing massive tissue infarction and hemoptysis.
Or Cryptococcus neoformans, the heavily encapsulated yeast that we inhale from pigeon droppings, which then travels directly to the brain to cause lethal meningoencephalitis in AIDS patients.
Both incredibly important pathogens.
But we will save the deep dives into Aspergillus and Cryptococcus for their own dedicated reviews.
The overarching takeaway for the opportunists is this. Always look at the host. The pathogen is just exploiting a specific crack in the armor.
Context is absolutely king. If a patient has profound neutropenia, your brain should immediately think Aspergillus.
If the patient has untreated HIV with a very low CD4 count and they present with a headache, think Cryptococcus.
And if a patient has been in the surgical ICU for 3 weeks with a central line and broad-spectrum antibiotics, think Candida.
The host defect defines the fungal risk.
Okay, we have traveled from the outermost surface of the skin, down through the hair follicles, deep into the subcutaneous tissue and the lymphatics, inhaled into the alveoli of the lungs, and disseminated completely through the bloodstream to every organ.
That was an absolute marathon.
It is a massive kingdom to cover. But if you mentally anchor yourself on the concept of depth of invasion, and you pair that with geography for the dimorphs, it all really starts to make logical clinical sense.
It becomes much less of a random list of bugs and much more of a cohesive narrative.
Let's prove it. It is time to lock this knowledge in. It is time for the rapid recall.
I love this part.
I am going to give you a short clinical scenario. Listen closely for the key buzzwords we discussed. I want you to visualize the patient, visualize the microscopic shape of the bug, and name the specific organism.
And listeners, please try actually say the answer out loud before I do. Force your brain to retrieve the information.
Here we go. Question 1. A 45-year-old man from rural Mississippi presents to the clinic with chronic cough, low-grade fever, and fatigue. A chest X-ray shows multiple calcified granulomas in the lungs. A biopsy of an enlarged cervical lymph node shows macrophages that are completely filled with tiny intracellular oval bodies. What is the organism?
Think about the clues for a second. We have Mississippi, and we have intracellular yeasts hiding inside macrophages.
Got it.
That is Histoplasma capsulatum. The geography points to the river valley, and the phrase histohides gives away the intracellular pathology.
Excellent. Question 2. A 6-year-old girl is brought to the pediatrician because she has a patchy, bald, very scaly lesion on her scalp. The doctor takes the child into a dark room and shines a Wood's lamp on her head. The hair shafts in the bald patch fluoresce a bright, vivid green. What is the most likely genus causing this infection?
Take a moment. Think about what causes the green glow.
Ready.
That is a Microsporum species. The bright green fluorescence indicates a classic ectothrix infection, where the fungal spores form a sheath around the outside of the hair, producing that fluorescent pteridine compound.
Perfect. Question 3. A pregnant woman in her 3rd trimester who lives in Phoenix, Arizona, develops a high fever, a dry cough, and very painful bright red nodules on the front of her shins. A lung biopsy reveals a massive, very thick-walled structure that is packed with hundreds of small endospores. What is the organism?
Analyze the risk factors: Arizona, pregnancy, and erythema nodosum.
Lock it in.
That is coccidioids. The clinical syndrome is valley fever. The painful shin nodules are erythema nodosum, and that massive structure on the biopsy is the classic spheral.
Question 4. An avid gardener presents with a painless, non-healing ulcer on her right thumb. Over the next 2 weeks, a series of hard red nodules appear in a linear pattern, tracking up her forearm along the lymphatic drainage pathway. What is the organism?
What's the mechanism of injury, and what's the distinct clinical pattern?
Go ahead.
That is Sporthrix synci. The mechanism is traumatic implantation from a rose thorn, and the diagnostic sign is that ascending lymphocutaneous spread up the arm.
Question 5. A microscopic exam of a skin scraping from a patient's groin reveals large, club-shaped macroconidia grouped in small clusters. No microconidia are seen anywhere on the slide. The patient has severe skin lesions and thickened nails, but perfectly normal hair. What is the organism?
Visualize those club-shaped spores. What do they look like?
Beaver tails, right?
Yes. That is epidermophyton flocosium. The club-shaped macroconidia and the absolute sparing of the hair are the key differentiators for epidermophyton.
Question 6. A patient in the intensive care unit with a central venous catheter develops a sudden spiking fever. Blood cultures are drawn, and they grow a yeast. When the lab incubates this yeast in animal serum at 37 degrees Celsius for 2 to 3 hours, it forms distinct germ tubes. What is the organism?
What bug forms that hand mirror shape in serum?
Vellum.
That is Candida albicans. The germ tube test is the classic, highly specific, rapid identification tool for this specific opportunist.
Last one, question 7. A 50-year-old male agricultural worker from rural Brazil presents with severe, painful ulcers on his gums and palate. A biopsy of the mucosal lesion reveals a large yeast cell with multiple buds protruding outward entirely around its circumference. What is the organism?
Think about the geography and that iconic visual shape.
A ship's wheel.
Exactly. That is Paracoccidioides brasiliensis. The South American geography, the male predominance due to the estrogen effect, and the pilot's wheel multi-budding morphology are absolutely diagnostic.
If you nailed all 7 of those, you're in phenomenal shape for the wards and for your board exams.
Truly, you understand the core concepts.
Now for consolidation, because active reading is the best way to make this stick permanently, I highly, highly recommend opening your copy of Murray's Medical Microbiology, specifically Chapter 59.
Yes, review the structured tables in Murray.
And pay special attention to the life cycle diagrams of the dimorphic fungi. Trace the path from the mold in the soil to the yeast in the tissue.
And as a supplementary visual aid, check Javits, Chapter 45. Look at the specific histopathology images. You absolutely need to be able to recognize the spherule of coccidioids and the broad-based bud of blastomyces on site. Visual recognition is a massive part of the exam.
And here's a final provocative thought for you to chew on as we wrap up. We've talked extensively about how dimorphic fungi transition from mold to yeast precisely when they encounter the 37-degree heat of a human body.
Right, it's their thermal trigger for invasion.
But consider the reality of climate change. Average environmental temperatures globally are steadily rising.
They are.
If the soil itself begins to regularly reach higher and higher temperatures, what happens to the evolutionary pressure on these fungi? Are we potentially selecting for strains that are already pre-adapted to mammalian body heat? Will the geographical ranges of things like coccidioids and histoplasma drastically expand as the climate warms?
That is a chilling thought. We are already seeing the endemic zones of valley fever creeping further north and west every year.
It suggests that the boundaries of the fungal kingdom are not static. They're dynamic, and they are moving. Fungi are not just mushrooms in the woods. They are complex, eukaryotic pathogens that ruthlessly exploit our geography, our hobbies, and the gaps in our immune system.
You really have to know the shape and the place to make the diagnosis.
That is it for our journey through the fungal kingdom. From the aliens quietly loitering among us to the invasive aliens destroying from within. Thanks for listening to The Deep Dive.
Keep learning. Goodbye.